Welcome to the second half of Module 1 on genomic selection, in which we were talking about breeding efficiency and the impact that molecular plant breeding can have on improving that efficiency. And here's just an example. In wheat, we have a disease called Fusarium head blight, a devastating disease, pretty widespread throughout the world, and we have one QTL in particular that's quite effective in improving resistance to it. It is a QTL. It does not provide complete resistance, and the name of that QTL is FHB1. And here's just a picture of Fusarium head blight in wheat. Here's a picture of a plot here. About 85% of those heads are showing symptoms of Fusarium head blight, and that's where the bleaching comes from. The next one, I would give that about a 15% rating. 15% of those heads and florets are showing symptoms of Fusarium head blight. And here's one that looks pretty resistant. You can't see it, but about 2% of those florets and heads are showing symptoms of Fusarium head blight. There is no complete resistance to this disease. It's quantitative, and again, this gene here is the most well-known one that has the largest effect. And that's shown over here. We created near-isogenic lines. Shown in the black bar are the performance of lines that have the resistant allele of FHB1, and the other one in the white bar is the near-isogenic line partner that lacks the gene. And here is our percentage of symptoms that we see. Without the gene, we're about 30% infected. Adding that one QTL in has taken us down to only about 7.5% infection. So a very nice, powerful gene that we use all the time in marker-assisted selection for Fusarium head blight resistance in wheat. How does this impact our efficiency of plant breeding? Well, here is my efficiency again that I showed before without any marker-assisted selection. But now let's say I'm going to do marker-assisted selection for FHB1, and now instead of only half my lines being acceptable for Fusarium head blight resistance, because of that marker-assisted selection, now 80% are acceptable. I take 3% times 80% times those others, and now instead of 1 out of 2,000 lines being acceptable, now 1 out of 1,200 lines is acceptable. To be 95% certain of finding such a line, I need to test about 3,700, and I can do that in about 3 to 4 years if I'm testing 1,000 a year. So that's a lot less time and a lot more efficient than if I did not do marker-assisted selection for that gene. And as long as I'm doing marker-assisted selection, now let's say I do marker-assisted selection for resistance to some of my other diseases. Now instead of only 10% being acceptable for resistance to all those diseases, now I'm up to 20%. I take 3% times 80 times 20 times those other numbers. Now my probability of the frequency of finding a new acceptable variety is 1 out of 600. If I test 1,800 of them, I would have a 95% chance of finding a new variety, and I could do that in 2 years if I'm testing 1,000 new lines a year. So my efficiency goes up quite dramatically from doing some marker-assisted selection for just a few major genes. And it just shows how this impacts your probability of success with certain breeding sizes. So again, 1 out of 2,000 if I do no marker-assisted selection. Marker-assisted selection for just FHB1, probability of a new variety, 1 out of 2,000. And if I do it for all those genes, 1 out of 600 is my probability. So no marker-assisted selection. If I want to be 95% certain of finding a new variety, I have to test 6,000 like I showed. And with marker-assisted selection for FHB, I now only have to test about 3,500, and marker-assisted selection for multiple genes, now I'm again about 1,800. So you can figure this out for your program, how efficient you are. Let's say you're testing only 500 new lines a year. Okay. And the probability of finding a new variety is 1 out of 2,000. You test 500 new lines a year. Your probability of finding that new variety is 22%, 0.22. If you do the marker-assisted selection, your probability of the new variety goes up. And if you test the same 500 lines again, now you test 500 lines again after marker-assisted selection, your probability of finding a new variety now is 34%. And if you do marker-assisted selection for all those genes, and you still test 500 lines, you're probably finding that new cultivar is now 57%. So if you're only testing the 500 lines, you're getting much more bang for your buck if you do some marker-assisted selection or molecular plant breeding on that material before it goes into your trials. Let's look at another way of doing marker-assisted selection, the genomic selection. This works for traits that are controlled by many genes with small effects like yield. So let's say I do genomic selection for yield. Instead of only 3% being acceptable for yield, now I'm up to 6%. That means 1 out of 1,000 could be new varieties. And I'd need to test about 3,000 of those to be 95% certain, and I can do that in 3 years. And basically by doing that, I'm twice as effective as if I did not do genomic selection. So what do you need to do what is needed for you to do molecular plant breeding? Well, first of all, you still have to be able to do your traditional plant breeding. And in particular, you need to be able to phenotype decent-sized populations accurately to get high heritability. Then, of course, you have to do everything else you do in traditional plant breeding. Plus, you have to do all these things that are in the box to the right. You need lab space, isolate DNA, you need a sequencer, someone to run the sequencer, bioinformatics, and the ability to analyze the data that comes out of the sequencer and interpret it into a breeding output. Some understanding of the statistics is nice. So when I first started doing molecular plant breeding, I looked through this kind of list and says, whoa, I really don't know how to do any of that stuff. Now I could train myself to do all that, but I'm a plant breeder. I'm busy doing this stuff in that box on the left. I cannot learn and operate all those things in the box on the right. So what you have to do is find a partner. Partner with someone who can do this for you. And that is really the main goal of the IGSS, is to provide all those kind of activities so that you can be a molecular plant breeder. And of course, IGSS is one place to get this. You might have many other people who can partner with and do this as well. But you will need a partner. So why do I want to use molecular plant breeding? Well, first of all, as we've shown, it can really improve the efficiency and my probability of success. I think I will get more bang for my phenotyping dollar spent. Better material enters my field trials. Better chance of finding a new variety for the plots that I run. And I'm a public plant breeder, and this allows me to better serve my public, my clientele. Directly addresses some of the key traits, helps me keep up with the competition. If all my competition releasing varieties is doing genomic selection, then I need to do it to stay up with them, or I need to change the focus of my program. Provides insights into traits and genetics, helps you with publications, helps improve your ability to get grants, you're staying current, and I just find it fascinating to learn about all aspects of plant breeding. So molecular plant breeding is, really helps you as you look at your flow of germplasm through your breeding program. This is something we could call the plant breeding funnel. At early stages you may have 1 million plants, F2 plants, but they're untested. You don't really know much about them other than their pedigree. Then there may be 100,000 might enter testing, 10,000 enter the next stage, 1,000 after that, and 10 lines end up to the last stage. Now when you're down at this stage of testing, you're actually getting all sorts of phenotypic data. Okay, so your selection based on phenotypes is pretty effective down here. But look up here at the top. Here's 1 million plants that have not been tested. They have no phenotypic data. These ones entering the first stage of yield testing. It might be just one plot in one location. You're not getting much phenotypic data there to help you with your selection. This is where the genotypic data comes in. Being the molecular plant breeder can really help you at the top of the breeding funnel. All right? No phenotypic data, but you can generate genotypic data and do selections. And of course, when you get down these later stages, the genotypic data doesn't help you much. You have years of phenotypic data. You know what your germplasm is. You pretty much know what it can do for you. So the goal of molecular plant breeding is sort of to broaden the top of your breeding funnel and actually kind of narrow the bottom of the breeding funnel. So instead of being shaped like this, you'll have this kind of shape. A lot more material is tested in this early stage using molecular technologies. That increases the effective size of your breeding program and improves your probability of success. Because of the molecular screens up here, better material enters your later stages of breeding and you get more value from all those plots that you run. So what do you need to get started? Well, you have to be a traditional plant breeder with accurate phenotyping, looking at decent sized breeding populations. You may be able to achieve a decent sized breeding population by collaborations. You have to have a relevant breeding population for releasing varieties and for the traits you're looking to improve. And you have to have a partners for some of these components down here. And you can't trivialize the fact that you need funds to get started. Okay, you need a willingness to learn, to partner with people, adapt what you do to accommodate molecular plant breeding, find ways to make it work. And a lot of that can happen by forming a community of practice and by collaborations. You learn from each other, you partner, you leverage your resources. IGS can help with a lot of these things. With that, I just want to close by saying plant breeding is inherently inefficient. Marker-assisted selection, genomic selection can improve your efficiency and thus your service to your clientele and return on investment in your program. There's many technical aspects to implementing molecular plant breeding, and it really helps if you have partners who can help with this. Here is a quiz. You can take this if you wish. You don't have to. If you do take it and you want to know how you did, just email me your answers and I will try to let you know. Thank you for viewing this material, and again, any questions, please contact me and I'll be happy to discuss.